Does anyone here like cilantro? Raise your hand if you do. Wow, that's a lot of yes. Okay, how about those that didn't raise your hand? Do you think it tastes weird? Smells weird? Yeah? Yeah. I see some nods and some confused faces, but it's okay. Because I'm about to tell you that for me, cilantro tastes like soap. Right? And cilantro is just one example where people have very polarizing experiences with the same food, but same thing also happens to coffee and broccoli. Some people can enjoy black coffee and also water-boiled broccoli, while some people only have coffee when there's enough sugar and milk, and also some sources for the broccoli to mask the bitter taste of it. So up to this moment, you may wonder, why is this happening, right? We are tasting the exactly same food, but have very opposite experiences. So then, thanks to hard work done by the geneticists, they found out for the cilantro dislikers just like us, we have a genetic variations in our olfactory receptor genes that makes cilantro taste like soap to us. And also lead to something that we have neglected before, but it should make sense if I tell you now that each of us perceive bitter taste differently. So how did the scientists find out this? It was found out firstly discovered in 1932, about 92 years ago due to a startling accidental discovery. So what happened 92 years ago is that two industrial chemists were preparing an experiment as usual, and then one of the chemists named Arthur Fox was dealing with a part of some sort of white powder compound called phenolphthalein carbamate, also known as PTC. So some of the PTC dust flew accidentally into the air, and his colleague said, "Oh, that is so bitter." Well, Fox is very confused because he tastes nothing. And both of them very, very confused, and then after further investigations and lots of people have tried it, they found out some people taste PTC intensely bitter, while some others taste nothing. So this is why they found out there are individual perception in the bitter taste perception. Traditionally, we know there are 5 taste modalities, such as sweet, salty, sour, bitter, and umami. And my research mainly focuses on the bitter taste. So bitter taste is innately aversive and is thought to as a warning signal to avoid us consuming toxins, which are often taste bitter. Well, after that discovery, the geneticists have found out there are many genetic variations in our bitter taste receptors, which have led us to have individual differences in the bitter taste. It makes sense, right? When we think about bitter taste, we're thinking about bitter taste receptor genes. But there could be other genetic variations in our genome that we haven't found out yet. So my research is to figure that part using a genome-wide association study. A genome-wide association study is an observational study that is trying to see is there any genetic variations in the entire human genome that is specifically associated with the trait that we're interested in. So in this case, in my case, it will be the bitter taste food preferences. I'm looking for is there any specific genetic variations that are specifically associated with the genome of those people that like bitter tasting food, or is there any genetic variations that appear more frequently in the genome of those who do not like bitter tasting food. To do this, I will use the current data from UK Biobank, which is a large-scale ongoing long-term biobank study comprised of 500,000 of volunteers in the UK since 2006. And with different aspects of data collected, you can see here, the UK Biobank is trying to enable to provide us with better understanding in the prevention, diagnosis, and also treatment of a wide range of diseases. So to do a GWAS study, I need the genetic data, and I also need the food preferences data, which were collected through the use of online surveys in this case. The genetic data is collected through the uses of microarray, which is a tiny plate you see here that can be within hold with our hand, but can assay around 800,000 genetic variations for multiple people at once. This is a very cost-efficient technology, and this technology made it possible for us to know our genes nowadays with the services provided by some genotyping companies. And then the food preferences data is collected through the use of food preferences questionnaire, which contains 144 food items, and the participants were invited to rate the degree of liking. So number one, 1 means they are extremely dislike this type of food, and 9 means they are extremely liking this type of food. And then I'll pick some well-conceived bitter-tasting foods from this questionnaire and construct a score for everyone who have rated their foods. Next, I will also do some quality control steps to make sure the accuracy of the data, and I'll run millions of statistical tests to see the result. And then the result will be plotted in a type of plot called Manhattan plot. The reason behind that is that it just looks like the skylines in the downtown Manhattan. And on the x-axis, it displays the entire human genome. We know that we have 22 pairs of autosomes in the human genome. So the number ranged from number 1 to number 22, and each block represents 1 chromosome. And there are many little dots within each chromosome. Every single dot in this map represents a genetic variation in our human genome. So overall, there are about 11.8 million dots on this plot. And on the y-axis is the p-values. So in statistics, the p-values is the probability of how likely the differences we see between those who like the bitter-tasting food and those who do not like bitter-tasting food is due to chance. So ideally, the smaller the p-value is, the less likely it's due to chance, and it's more likely the true differences we see. So in the GWAS study, the p-value is usually very small. It's smaller than 5 times 10 to the power of -8, which is very small and very hard to visualize. So I'll take the negative log of the p-values so that those with the smallest p-values will appear on the top of the figure. And that's the part that I'm interested in. And those are the genetic variations that are associated with the trait I'm interested into. So are you ready to see the real plot? Yeah? So here is the plot. It's the plot for the association between a group of bitter-tasting foods comprised of black olives, green olives, grapefruit, and also the bitter-tasting food. So like I said, the interesting part is highlighted within this red box. We can see that there are some genetic variations that are very strongly associated with the food I just mentioned, and they are from chromosome 2, chromosome 3, chromosome 7, and chromosome 12. And after that, I annotated these genetic variations to the genes they are belonging to, or to the nearest genes they belong to, and I found those are the genes that they are coming from. It's not surprising that I see some TAS2R genes, which stand for taste receptor genes. That makes sense, right? The bitter taste receptor genes. But there are also some other genes that are associated with the personality traits or associated with the development of forebrains. You may wonder, how is that possible, right? The bitter tasting food and the personality and also the development of forebrain. That's a very good question, but I don't have an answer for it yet. I'm leaving that question to the future students and researchers, and maybe just one of you guys. So how can we use the result from this study? It will lead us to a better understanding of the bitter tasting food genetics. And remember, there are also other taste modalities like sweet, sour, umami. We can also apply the same thing to those, and that we will have a better biological insight in the food preferences. And hopefully one day, with all those knowledges, we can use them in the development of personalized nutrition. So thank you very much for your attention, and to special thanks to you and in the whole process of helping us to develop the presentation. And I'm happy to take any questions.